Hello and welcome back to Health Unlock, the Power of Salutogenesis with Jasen Petersen. Jason, how are you?
I'm doing pretty good. As we were discussing, there's a little bit of snow around, but it's also not that cold. And actually, for the next few days, I think it's going to be in the 40s and 50s. Well, there's a lot of snow. We'll see how quickly it disappears. Going to be slushy around for a bit, I think.
Yeah, let's know. But you're looking fresh in your fresh haircut, so that's good. You're ready for the weekend. That's awesome. And you're ready for today's episode, which is going to be, I think, one of the most fascinating of our all that we have done up until now. So jumping right straight into the topic, today we are going to talk about cell suppression theory and a lot of things about cancer and a lot of things that we want to uncover, unleash, but also educate people on. Um, all right, let's jump in.
All
So
right.
most people accept the idea that cancer is caused by some genetic mutation, but you don't subscribe to this and you don't subscribe to the somatic mutation theory. Why not? And what do you think is wrong with it? And why do you believe so many people are, still remain committed to this old model or the belief?
Yeah. Um, so I guess I want to start by saying Mark Lintern details a lot of this in his book, the Cancer Resolution. And so rather than referencing every time I say something that's from his book, I'm just going to say up front that I'm going to pull lots of references and things from his book. Um, but so and he goes through a lot of this in his book as well. So the somatic mutation theory states that cancer is caused by accumulated DNA mutations that lead to uncontrolled growth. But if that were true, we would expect a predictable pattern of mutations across all cancers. We would expect the same driver mutations to be present in all cases of a given cancer. And then we also would expect a direct causal relationship between the DNA mutations and tumor formation. But the evidence kind of contradicts these expectations, making the somatic mutation theory, or SMT, an incomplete or even misleading explanation of cancer. So I've got a bunch of reasons. So I'll try and rattle them off pretty quickly, but hopefully they don't get too involved. The first one really focuses on the hallmarks of cancer. So doctors Hanahan and Weinberg, they put together what's known as the hallmarks of cancer. And it started off, I think, with just 6, and now it's at 10, and they might add some more to it. But basically, the idea is here are these 10 things that always occur with cancer. And if you're either trying to explain what cancer is or to treat cancer, if you can explain, if you come up with a theory and it explains all of these things, all 10 of these things, then likely you're pretty close. And or if you're treating cancer, well, if you can treat all 10 of these things, then likely that's a good treatment. So it gives you a really good framework from which to examine this, right? So let's look at them. There's uncontrolled cell growth, resistance to anti-growth signals, evasion of cell death signals. Those all kind of sound like the same thing, but they're a little bit different. Limitless replication, extended blood vessel growth, invasion of local tissue and metastasis, the abnormal metabolic pathways or the Warburg effect, the evasion of the immune system, genomic instability or DNA damage, and inflammation. So, of all of those, SMT was expected originally to explain 4 of them. 1, uncontrolled growth. And it was expected that the mutations in K, ROS, and APC are specific genes that were thought to turn on growth signals permanently. And it actually fails because many cancers actually lack these mutations, but they still grow uncontrolled. Then you've got the number 2, I guess, is resistance to those anti-growth signals. So you get the reason it was supposed to explain this was the loss of the tumor suppressor genes, APC, and then the P53, or I guess the gene is TP53. P53 is the protein that it encodes. So those were assumed to remove the growth brakes. But again, this fails because some cancers retain functional tumor suppressor genes, yet still ignore the growth signals. Next one is the number 3, so evasion of apoptosis. And the thinking was that TP53 mutations prevent the P53 from activating apoptosis. And that's still kind of valid because about a third of all cancers do have TP53 mutations. So I guess we can give them that one. Number 4 is limitless replication. So the telomerase activation was assumed to be due to genetic mutations, but again, fails because many cancers, they extend replication through metabolic shifts rather than the genetic changes. So that one fails as well. And then the last one that it was supposed to explain was genomic instability. And I guess SMT still explains DNA damage. SMT was supposed to explain that how DNA damage accumulates over time. And that remains true, right? So cancer cells do accumulate DNA damage, but this appears to be a consequence and not a cause. So, summarize all that. This somatic mutation theory only explains some of the hallmarks. Specifically, was genome instability, hallmark 9, and resistance to cell death, which is hallmark 3. It fails to explain why all the cancers exhibit the Warburg effect, hallmark 7, and why immune evasion is universal, why chronic inflammation is a common precursor. So basically, let's back up for a sec. If DNA mutations were the sole cause of cancer, and then the SMT should account for all of the hallmarks, not just a fraction of them. So that alone, all by itself, suggests that there's another factor, right? That beyond random mutations that's driving cancer. That's the first problem. Second problem is it actually came from the Cancer Genome Atlas project. And so that was expected to prove that specific DNA mutations consistently drive cancer. Instead, it kind of found the opposite. So after analyzing over 13,000 cancer cases, only about a third of them showed that they had that P53 mutation. Remember, that was what was one of the things that was expected. Now, the expectation was that you needed to see at least 60% of cancers were going to show that mutation, but really it's only a third, so about half of what it was expected. I think so. A study of like colorectal cancer found it was like something like 6.6% of the samples contain mutations in all 3 of the essential genes. Those essential genes were, I mentioned before, the TP53, that KROS, and APC. So TP3, KROS, and APC, they were considered the essential cancer genes because they regulate 3 key processes, right? Apoptosis for P53, growth signaling for KROS, and then tissue organization or tumor suppression for the APC. Some tumors had no mutations in any one of those genes. So you would think that these results from the cancer genome study should have been a death blow to the somatic mutation theory, but it wasn't. If SMT is correct, then all cancers should contain the required driver mutations, right? This kind of makes sense. And instead, the data is showing that even if we look at so-called essential mutations, many times they're absent. So how can DNA mutations cause a disease if the mutations aren't present, right? Yeah. So that's problem 2. Problem 3. And this one, I'll just kind of go through real quick, because this one I truly took directly from Mark's book. I actually hadn't thought about it before. Obviously, somebody else had. But the mutations actually take too long. So there's something of a paradox here. So the SMT suggests that cancer, takes decades to develop because it requires at the time, it was like 10 to 12 driver mutations to occur. And here you'd have to know all the probabilities and everything. So I'll just take it for face value. But the theory kind of collapses under basic probability, right? So if cancer requires these 12 sequential mutations, the mutation rate is far too slow for that to happen within a human lifetime. But we know that it's happening. And I think it was Professor Larry Loeb that first questioned this. But I'll move off of that real quick because, well, what if mutations are just happening a lot faster these days? Okay, maybe. So number 4 then, problem number 4. The DNA mutations are somewhat random, but cancer is very predictable, right? So if random DNA mutations were responsible for cancer, we would see a consistent set of those driver mutations, right? In every type of cancer. But instead, cancer behaves in a very predictable manner based on its metabolic and immune environment, regardless of mutations, right? So you can have all sorts of different mutations, but the same thing kind of always happens. And, so cancer shares all these common traits, including the metabolic dysfunction, I'm putting that in little finger quotes for now, the Warburg effect, and immune evasion. And these patterns they can't be explained by random genetic mutations, right? So other genetic diseases caused truly by DNA mutations are highly consistent. For example, sickle cell anemia is caused by a specific mutation in the HBB gene every single time. But in cancer, there's no consistent mutation pattern. Instead, mutations appear random, even within the same tumor. So that suggests that mutations aren't the cause; they're a symptom, right? So run through all that real quick. Why do I believe that somatic mutation is wrong? It only explains a fraction of the hallmarks. The Cancer Genome Atlas shows that many cancers don't have the required mutations, or what was expected to be required mutations. The mutation rate is too slow to explain how it develops, and the mutations are random, but cancer follows a predictable pattern. That's why I'm fairly convinced SMT is wrong.
So if mutations aren't the root cause of cancer, what is? I guess we've discussed mitochondria a lot, and we're going to guess that you're more aligned with the metabolic theory of cancer. Am I right? Do you want to comment on that?
Yeah. So let's run through that real quick. So the short answer is yes, I am much more aligned with metabolic theory of cancer. So what is that? In the 1920s, Dr. Otto Warburg discovered that cancer cells produce high lactate levels even in an oxygen-rich environment. Basically, they're in glycolysis. And he hypothesized that mitochondrial damage was actually the root cause of cancer then, forcing the cells into glycolysis. Now, so most people actually believe that the Nobel Medicine Prize that he received in 1931 was tied to the discovery that we call now the Warburg effect. It's his name, right? But he actually received it for identifying that cytochrome C oxidase, if you remember that, the enzyme that transports oxygen into mitochondria in complex 4 of the ETC or electron transfer chain. So I guess if we move in time real quick to the 50s through 70s, this is when DNA mutations were discovered in cancer cells, and that kind of shifted the focus toward the somatic mutation theory. So the Warburg's theory kind of was mostly ignored after that, in favor of the genetic explanations, right? And then it was in the 2000s to kind of present, there's sort of this mitochondrial or metabolic theory of cancer resurgence, and studies are kind of starting to confirm that cancer cells have abnormal mitochondrial function and metabolic and the metabolic reprogramming that's involved in all of this. And then, you have more researchers that are, I think it's Thomas Seyfried, that are renewing interest in this, arguing that metabolic dysfunction is the primary driver of cancer. But so we use the hallmarks of cancer to show that SMT's failure, right? So let's do the exact same thing and try and use the hallmarks of cancer to explain why the metabolic theory is more right. So again, first one was uncontrolled growth, right? So what's really happening there is cells are prioritizing survival over regulation. And this the dysfunctional metabolism traps the cell in the proliferative survival mode, or CDR2. That explains why that you have the uncontrolled growth. The resistance to the anti-growth signals, you've got the energy imbalance overrides checkpoints. So metabolic or chronic metabolic dysfunction alters growth factor signaling, overriding normal suppression mechanisms. So that one's explained. You've got evasion of apoptosis, right? So mitochondria fail to trigger the cell death. Well, if the mitochondrial are dysfunctional, then they can prevent proper apoptosis signaling. That's fairly straightforward. 4 is the limitless replication. And again, so cells that are locked into that persistent proliferative state, and mitochondrial dysfunction would prevent the transition from CDR2 to CDR3, keeping cells in that repair proliferation state. Remember, we've talked about how the mitochondria are kind of the initiators of, and I have to be careful about how I say this because I'm going to clarify this a little bit later, but they're the ones that follow through on the phase shifting from CDR1 to 2 to 3 and so forth. So now, that one is explained. What are we on? We're on 6. So invasion and metastasis. So you've got energy stressed cells. So they actually have to migrate to survive, right? So the metabolic dysfunction creates a hostile environment with, reactive oxygen species and lactate buildup and excess iron. And that actually forces cells to migrate to search for better conditions so that they can persist better. Does that kind of make sense?
Yeah.
Okay. 7 is the actual Warburg effect, or aerobic glycolysis. So there's the shift in energy production, it's not really an increase in glucose use. So the cause of the metabolic and mitochondrial dysfunction was actually historically unknown, but its occurrence was well documented. So, ironically, the damaged or dysfunctional metabolism, which is hallmark 7, that's the one that the metabolic theory can't explain by itself, and that's the only one that it can't explain by itself. So, moving on, number 8 is the evasion of the immune system. Well, these metabolic adaptations suppress immune recognition, right? So you've got chronic metabolic stress driven by the excess reactive oxygen species, lactate buildup, and dysfunctional mitochondrial signaling. That all disrupts macrophage function, weakening the immune response. You've then got reactive oxygen species overproduction or ROS overproduction. And in a persistently glycolytic state, that then pushes macrophages into an immunosuppressive or exhausted state. You get lactate accumulation, and that also plays a role by altering macrophage polarization and dampening inflammatory responses. 9 is genomic instability. Well, that's not the cause. That's just a consequence of the chronic oxidative stress, right? So the cells are continually producing reactive oxygen species due to the mitochondrial dysfunction because they're not operating in oxidative phosphorylation or OXPHOS. And therefore, you get lots more reactive oxygen species that can damage DNA. Okay, that one's pretty straightforward. And the last one was the tumor promoting inflammation. So, right, a byproduct of metabolic stress, exactly what that is. So you get the metabolic dysfunction creates the chronic inflammatory state that then reinforces cancer progression. So, metabolic theory of cancer explains all of the hallmarks of cancer pretty darn well, except for the one specifically about why does the metabolism shift. That's the only one it can't explain by itself. So, yeah, I think that one is far more correct than SMT is.
So it should be always the dominant theory all along for this explanation. But why do you think so many people still remain committed to SMT?
You're right. So yes, it should have been the dominant theory all along. But why was the SMT adopted over it in the first place? We kind of have to look at the history here a little bit to understand why SMT gained traction. So let's, it actually, to be honest, it started with some mistakes that Otto Warburg made. So back in the 1920s, he observed that cancer cells were relying on glycolysis even when oxygen was present, right? So that is the Warburg effect. But he hypothesized that that it was mitochondrial damage that forced that metabolic shift, making cancer a disease of energy production. And so really that was his mistake, right? So he insisted that oxygen utilization was the primary defect, not mitochondrial signaling. And so when researchers later found that mitochondria in the cells, in cancer cells, could still use oxygen, they just dismissed his theory altogether. Now, had he framed it as a mitochondrial signaling disorder rather than just an oxygen utilization problem, it might have survived. But so then there was also a lot of other things going on at the time. In the 50s, the DNA's role of cell function kind of became clear. That was when Watson and Crick discovered the DNA structure 53. I think that laid the foundation for genetics-focused cancer research. Then in the 70s, they discovered those oncogenes and the tumor suppressor genes that KROS was the oncogene and the TP53 was the tumor suppressor, and they were found to play roles in cancer development. And this gave researchers kind of a tangible genetic target for drug development. And then gene-focused research was actually easier to study. And then was more commercially viable, making it more attractive for funding than metabolism research. And I understand that sounds weird, but the tools for studying DNA, like DNA sequencing and stuff, that was advancing pretty rapidly. Whereas at the time, mitochondrial research was much harder because the metabolic complexity made it really difficult to study. And so, once cancer was framed as a genetic disease, SMT became, then the dominant model simply because it's easier to study that with the existing technology. And I realize that's a really bad reason, but that was kind of the reason. And And, then you also have to remember that SMT then kind of fit the scientific trends of the time, right? So the 50s through 70s, the central dogma of molecular biology really was, you've got DNA, which leads to RNA, which leads to proteins, and that became the focus. And so the idea reinforced the belief that DNA mutations must be the origin of diseases, including cancer, right? So, and as molecular biology kind of exploded in popularity, making gene-focused research this real hot topic. And by contrast, metabolism research seemed outdated, and Warburg's ideas kind of were largely forgotten. So really, kind of the rise of molecular biology made SMT kind of fit the dominant scientific narrative. Another thing that you have to think about is the medical industry is really built on DNA-based diagnoses and treatments, right? So billions have been spent developing genetic testing, targeted therapies, these mutation-based drug models. And if cancer is metabolic, many current cancer treatments, chemo, immunotherapy, targeted drugs, and all this stuff, they'd all be kind of obsolete. And the whole medical industry is not designed to pivot quickly. There's too much invested into the genetic model, right? Then you also have to look at just education, right? So scientists have been trained to view cancer through a genetic lens. The education system trains researchers to assume DNA mutations cause cancer, reinforcing the genetic paradigm right from the start. And research funding then reinforces this bias. Labs studying genetics get the most grants. So MTC, or metabolic theory, is often dismissed not because it's wrong, but because it doesn't fit the dominant scientific or academic narrative. And that's, again, really bad reason, but that's the reason. Scientists are discouraged from questioning the SMT, that can end careers. Funding, tenure, and research opportunities are all tied to, genetic research. And so what was it? Consensus science, right? It kind of prevents open debate. I believe Mark actually quotes this in, so it's Michael Crichton said this, and Mark quotes it in his book. If it's consensus, it isn't science. If it's science, it isn't consensus. Crichton, the quote actually goes on to say that consensus is the business of politics. But in the end, really, few scientists are willing to risk their careers, funding, reputations, and all that, challenging the dominant model. Now, you should argue, the argument should be, well, why did the Cancer Genome Project kind of correct all of these mistakes, right? Because that Human Genome Project was in the 90s to 2000s. Actually, a good friend of mine was working, making devices to help with the gene sequencing and all that kind of stuff. But the idea was that scientists at the time, they believed that by sequencing the cancer genomes, then they'd find out the exact mutations driving each cancer, and then they could work on that and fix them. And so it was in 2005 that Cancer Genome Atlas project was either started or finished. They found that there was no single set of driver mutations across cancers. Some cancers had none of the essential mutations. And instead of abandoning it, the theory, they just kind of redefined what driver mutations meant to kind of fit their data. Again, this just reinforces it was too big to fail. A lot of this also is driven by big pharma, and funding structures and everything, right? Once SMT took hold, all funding, research, and drug development was built around genetics. Pharmaceutical companies saw massive profit potential in the gene-targeted drugs. And metabolic theory doesn't really lend itself to high-profit drug development. lifestyle, diet, metabolic interventions. Those are the best ways to work with the metabolism, and they don't make millions. So, yeah, I guess the decades of research funding poured into DNA cancer or DNA-based cancer therapies is too much to at least too quickly turn around. I feel like if I keep on going here, I'm just going to keep saying the same thing.
So, okay, so the MTC explains nearly everything, all the points except obviously the Hallmark 7, the Warburg effect. But we have discussed the CDR a lot, the cell danger response. So, and you have mentioned it, like the cells are stuck in CDR2, and that's why, that's why the mitochondria prioritizes the aerobic glycolysis there. But before we go and discuss that, can you do just like a refresher of the CDR one time, and then we can go to the follow-up question? Yeah.
Yeah. Okay. So remember, the cell danger response, or I'll probably say CDR a lot, is the body's built-in defense system. Right. So it's how cells react to danger signals, DAMPs and PAMPs, whether from injury, infection, toxins, radiation, other threats, whatever. Let me because I'm getting acronym heavy. Let me explain what a DAMP and a PAMP is. Right. So DAMP is the damage-associated molecular pattern. It sounds all fancy. It's really, these are just cellular components that belong inside cells. When they end up outside the cells, at least in large amounts, it's a clear signal that some cells have been damaged or destroyed or, ripped open so that the stuff on the inside is now on the outside. That's all a DAMP is. Now, a PAMP is just a pathogen-associated molecular pattern. And that's just, that actually, conversely, that comes from pathogens. And those are things like bacterial cell wall fragments or viral RNA or fungal byproducts, whatever. This is, so basically the immune system isn't supposed to see those things inside your body at all. And so when it does, it's an even bigger or clearer threat than DAMPs are. So either way, both act as alarm signals. Those are the alarm signals that we talk about a lot, that tell your immune system that, hey, there's a problem here, do something about it. And as we discussed, the cell danger response models, it details this process in 3 phases, 3 steps, right? First one, super fancy naming of CDR1. That's the immediate defense mode. So that's, you get the inflammation becomes active, and M1 mitochondria is dominant. Remember that M1 mitochondria is where they shift. They no longer operate in oxbox, and they shift to producing reactive oxygen species to combat the threat, right? So cells, in this phase, cells stop normal function, and they prioritize inflammation and immune defense. The mitochondria then do their shift. They start producing reactive oxygen species to combat the threat. Glycolysis becomes the main energy source. And lots of times you will see that it's faster. Well, not really. I mean, yeah, it's faster, but glycolysis is always happening. That's just happening in the cells. It really doesn't, the mitochondria don't have to do anything. That just happens. So when people start saying that, oh, mitochondria shift to glycolysis, well, they don't. They just stop doing oxphos. They stop increasing the efficiency so that they can focus on immune signaling and or other things, right? So the cells start releasing those inflammatory signals, remember the DAMPs, and that activates the immune system. Iron becomes tightly sequestered to keep it away from the pathogens. Apoptosis is inhibited to keep as many functional cells engaged in the response as possible. Now, so here's, if cells fail to exit CDR1 for whatever reason, you can run into chronic inflammatory and autoimmune disease problems, like rheumatoid arthritis or inflammatory bowel disease or psoriasis or multiple sclerosis. Next phase is CDR2. That's the repair and proliferation phase. The M1 phenotype of the mitochondria starts to decrease and M0 increases. So really, as the immediate threat subsides or adaptive immunity starts taking over, doesn't mean that the threat is gone, right? It's just that the reactive oxygen species you've been creating tons of, that gets to die down a little bit, and your adaptive immune system comes in and starts taking over. So the cells themselves can stop focusing purely on defense and then start transitioning to into CDR2 for repair and recovery. So the mitochondria, again, it still maintains glycolysis. That's the wrong way to put that, right? Because it's not doing anything with glycolysis. Glycolysis just happened. But so the Warburg effect continues here. And because the mitochondria are prioritizing rebuilding over energy efficiency, even when oxygen's available, right? So in this stage, reactive oxygen species start to drop a little bit, but small amounts remain because that's the signal to initiate and or continue tissue repair. Apoptosis remains inhibited. That allows healing of damaged cells, right? You want to do all the healing first before you go through and remove what you don't want. Iron actually starts to be released again. And that actually supports mitochondrial recovery, because at some point here, the mitochondria are going to have to shift back to M2, which is the one that we consider normal, right? So what happens if cells get stuck in CDR2? Well, then you can run into the chronic proliferative diseases, including fibrosis or polyps or benign tumors or cancer. And then CDR3, last phase, that's resolution and reintegration. M2 starts to increase or returns as the phenotype for mitochondria. Homeostasis gets restored. So mitochondria is now starting to restore oxphos, returning to normal energy production. The reactive oxygen species levels drop even more. Inflammation starts to resolve. Immune signals return to baseline. Apoptosis resumes, right? So now you want to remove any cells that haven't been able to fully recover. Iron metabolism normalizes. And so now that will support proper full mitochondrial function and tissue recovery. Now, so if cells fail to exit CDR3, you could get like lingering dysfunction that persists, leading to persistent infections and degenerative diseases like Alzheimer's or Parkinson's or osteoporosis. So I added a little. I tried to recap everything, but I added a little bit to it so we can kind of understand what happens when you get stuck in each one of those stages. I know we've kind of discussed that a little bit, but it was really high level. So hopefully I could add a little bit more detail there.
Yeah. So based on the explanation itself, so I'm convinced that cancer cells are stuck in CDR2, but you haven't explained why or how that happens. Like, why are they stuck there? And it feels, can you elaborate on that a little bit? Like, why, what exact phenomena there?
So this is the crux of Mark Lintern's cell suppression theory, right? And it builds upon Dr. Navio's cell danger response model to do exactly that. Explain why some of these cells, or why some cells get stuck in CDR2, and instead of progressing to CDR3 and healing. And he lays out all of this in his book, the Cancer Resolution, really well. I encourage anyone who's interested in this to purchase the book and read through it. It's pretty good. So really, we've kind of got just a few steps here. The first step is repeated activation of the CDR creates a terrain that becomes more advantageous to microbial infection than not. So cancer doesn't develop instantly, right? It emerges over time, primarily due to this repeated activation of the CDR. Each time a cell enters the response, it undergoes that metabolic shift, prioritizing survival over normal function. The longer this the cycle continues, or the more often you go into it, the more the local environment shifts away from health and into dysfunction, right? And this really, so carcinogens, really any persistent stressor that, toxins, infections, chronic inflammation, radiation, hypoxia. So, if you're not getting enough oxygen to some cells, or long-term stress, all of these things keep triggering the CDR. And as a result, you get iron. So the body starts to get worse at being able to regulate the iron. You get oxidative stress buildup, and you get metabolic imbalances. They increasingly become pronounced, right? And cells stuck in this cycle rely heavily on glycolysis. We've talked about this a bunch. And as a consequence, lactate and iron accumulate. So Mark points this out more than anything else, I think. And while those shifts were initially meant to support survival and healing, they can have unintended consequences and effects. And in this case, certain pathogens, particular fungi, can exploit the altered terrain. Lactate and iron, which should have been carefully regulated, instead become fuel sources for microbial persistence. So the second step is now, cells don't get stuck in CDR2 on their own, at least not in this model. There's an external factor that's actively preventing them from transitioning into CDR3 or resolution. And, as the CDR activation continues and the terrain shifts away from health and towards fungal colonization here, right? So, as I mentioned, iron regulation breaks down, leading to excess iron available. Lactate continues to accumulate as glycolysis remains the dominant metabolic pathway. Cell membranes become more permeable, and that's an adaptation to facilitate immune responses, but one that also leaves cells more vulnerable to microbial invasion, right? Because, they're porous. And the immune system or response is weakened, further reducing the body's ability to detect and clear infections. So now at this point, the fungi can exploit the terrain advantage and enter cells. Now this is, Mark isn't convinced that the fungi needs to be intercellular. I kind of am. I'm just going to go with it for now. So once the fungi gain intracellular access, they don't just persist there, right? They start to actively manipulate immune signaling to maintain their advantage. So some fungal species produce bioactive molecules that suppress apoptosis, preventing the cell from self-terminating when it should. Other fungi can interfere with mitochondrial function by altering oxidative stress pathways, keeping the cell reliant on glycolysis rather than shifting back into oxygenase. And I guess beyond the individual cell level, fungi can also suppress immune responses system-wide. Some species produce active compounds that reduce antigen presentation, dampening the adaptive immune activation. And this is one of the reasons why fungal infections can be so difficult to clear and treat. Because unlike bacteria or viruses, they've evolved really elegant ways to hide within host cells, while simultaneously weakening the cell's ability to respond. Let me jump to the third step here, right? So once fungi have been able to kind of now hijack the immune system signaling and prevent CDR resolution, the cells remain trapped in that survival mode. And then their metabolism's locked into glycolysis. Their proliferation continues unchecked, and their immune environment becomes increasingly suppressed. And it's really at this stage that, we kind of see that the hallmarks of cancer begin to emerge, right? We already know cells are remaining persistent in that CDR2 state, where they replicate, continue replicating, and instead of resolving, they maintain the glycolytic metabolism of the Warburg effect, which supports growth over energy efficiency. They develop genomic instability due to the continued oxidative stress and metabolic dysfunction, although, as we pointed out, that's not really metabolic dysfunction. That's just the mitochondria not functioning to make energy, but to make reactive oxygen species. And you also, I guess they fail to communicate correctly, or the cells failed to start communicating correctly with the immune system, and that leads to the formation of then the tumor microenvironment. And now at this point, everything we associate with cancer, from that uncontrolled growth to immune evasion and all that kind of stuff, emerges as a direct result of the cells being trapped in CDR3. Normal signaling is actively being suppressed, but it's also passively not very good. And as long as the suppression continues, they can't really transition to CDR3 and complete the whole healing process. So it all comes down to fungi infect cells. Sorry, apparently I didn't turn my phone off here. There we go.
Yeah. So now we have discussed this before, how mitochondria and oxygenation play a critical role in the health. How do these factors connect with the suppression theory's explanation of cancer?
Okay, so the mitochondrial function and cellular oxygenation, they're not just kind of abstract health concepts here. They actually play a central role in the CST and how cancer emerges, right? So their function is critical at three levels, I guess. Right? So you've got maintaining metabolic health, regulating immune function, and controlling CDR progression. That's kind of the one that we're talking about a lot here. So if mitochondrial function is compromised, and it doesn't, remember, it can be compromised by all sorts of any of these things occurring. The body then struggles to respond to threats, regulate inflammation, and maintain resilience. So the function or the dysfunction doesn't just affect systemic metabolism. It impacts local tissue environments and immune cells, including macrophages, which help orchestrate the immune responses and regulate iron sequestration. Is that how you say that? So when mitochondrial function declines in these key players, their ability to regulate CDR progression weakens. So that's really important. And this can lead to disrupted immune signaling and prolonged survival states in stress cells, making it easier for them to be trapped in CDR2. Actually, easier to be trapped in any one of the phases, but if we're really talking about cancer, trapped in CDR2. If we talk about metabolic flexibility, right? So that's the ability of mitochondria to shift between different fuel sources like glucose and fatty acids efficient. And it directly affects the body's ability to regulate the CDR. So I guess most people today are too carb adapted, meaning that our mitochondria primarily rely on glucose. That can then reinforce this glycolysis dominance and make it harder for cells to exit CDR2 and return to oxphos, whether through whatever, a ketogenic diet or other metabolic strategies, encouraging the fat oxidation can help shift, regardless of what your strategy is. Encouraging the fat oxidation can help shift mitochondria back towards oxphos, improving overall resilience and cellular recovery. And then beyond just the fuel flexibility, you've got oxygen availability. That's another key factor that determines mitochondrial function, as we've discussed before. Oxygen is required in oxphos, which produces significantly more ATP than glycolysis. And if oxygen delivery is insufficient, either due to local hypoxia or mitochondrial dysfunction, true mitochondrial dysfunction, the cells, they default to glycolysis because they're doing that anyways, right? And then they remain in that pro-survival state. And this is where, the systemic oxygenation strategies like exercise oxygen therapy, post-electromagnetic field therapy, and photobiomodulation can support mitochondrial efficiency and, ensuring oxygen isn't the limiting factor for energy production. And I guess I want to point out that this metabolic flexibility or mitochondrial function and of course adequate oxygenation are particularly important for macrophages because they need to rapidly transition between they have and unfortunately it's the same nomenclature. But they've got different phenotypes that they shift into in the same way that mitochondria do. But remember, mitochondria are also inside the macrophages. So they have to quickly shift between their M1 phenotype, which is inflammatory, and their M2 phenotype, which is the resolution, those states to regulate immune function, right? So the M1 macrophages are pro-inflammatory, rely on glycolysis, just like cells in CDR1. M2 macrophages are anti-inflammatory and depend on oxphos, just like cells in CDR3. And I guess this would be weird if I don't then say we also have M0 macrophages, which are neither pro-inflammatory nor anti-inflammatory. They exist in a neutral state and then can differentiate into either M1 or M2, depending on the signals that they're receiving from the environment. In the context of CDR progression, they often correspond to CDR2. Where they help regulate the tissue repair and immune adaptation. So improving systemic mitochondrial health and oxygenation is a great starting point to treat cancer, right? It enhances resilience and immune efficiency and cellular recovery and all that. All that's great. But more often than not, it's not enough on its own.
Why is it not enough on its own?
Okay. Sometimes it is, right? So sometimes it's enough. Sometimes just stimulating the immune system is enough to sidestep that fungal control of CDR progression. And sometimes that's even enough to then eliminate the fungal triggers entirely. But unfortunately, not nearly as often as we'd like. And that's because systemic metabolic health alone won't always fix a localized suppression problem, right? So you can have perfect systemic, you can have really good systemic metabolic function and still develop cancer if local immune fatigue remains, right? And so cancer really is a local first problem. Many people confuse systemic and local immune fatigue, or they've never even thought about it. So you can have a strong systemic immune system and still develop cancer. I saw this firsthand with my parents and sister. They rarely ever got sick, but they all developed cancer. And actually, even with my sister, while she was healing, she was objectively healthier than almost everyone I knew, except from, the whole thing that she had cancer. But you would never know it because when everyone else was getting sick with colds and flus and that kind of stuff, she didn't. So her systemic immune system was really good. So that's, I guess it's because, so obviously it can be a contributing factor, right? But cancer doesn't develop due to a weak systemic immune system. It starts with local immune fatigue. So there are multiple components of the local innate immune system that when chronically activated, the terrain advantage shifts to fungi. So some of those things, you've got chronic immune activation increases epithelial and endothelial permeability, making it easier for microbes, including fungi, to invade, especially in high lactate environments. You've got mast cell overproduction, basically persistent inflammation. And that releases excess histamine and inflammatory mediators. What that does is that increases the vascular permeability and depletes the immune reserves. You've got dysregulated innate lymphoid cells, ILCs, and that can drive fibrosis. It basically prolong inflammation instead of driving proper tissue repair, and further weaken the epithelial barriers, allowing fungal colonization. So really, the repeated activation or activation of those components doesn't just weaken the immune responses, it actively shifts the terrain towards fungal persistence. And we mentioned macrophages. I want to kind of dig into this a little bit more. So even though there are lots of components, there are, a bunch of key players here involved in the immune system, their actions are more singular and reactive. Whereas macrophages, they're a little different. They are, you can consider them the immune system multitaskers. They're responsible for detecting threats, coordinating immune responses, and maintaining tissue homeostasis, right? So they detect and respond to threats, those PAMPs and DAMPs, and then they trigger inflammation. They engulf and destroy pathogens using reactive oxygen species and enzymatic digestion and stuff. They activate and direct other immune cells. So they present like antigens to T cells to recruit neutrophil and dendritic cells. They clear dead cells and repair tissue. And so they suppress inflammation, and they their M2 state, and then promote tissue remodeling. They maintain immune surveillance. They regulate the cell danger response. So they signal structural cells to enter or exit CDR phases, sequester iron, and coordinate immune transition. So I might have buried the lead there, because regulating the CDR response is what makes them really important for our discussion. And I want to, so macrophages come in two types, right? The monocyte derived macrophages. That's what we normally think of macrophages as, they circulate throughout the body and then respond where they're needed. But then there's also tissue resident macrophages. So they stay embedded in tissues for their entire life cycle, with some minor, almost all the time. And so what that means is that chronic activation leads to local metabolic fatigue there. They're the ones that we're most interested in, right? So let's talk about their TRMs, right? So tissue resident macrophages. And they play a really pivotal role. I actually would argue that in the cell danger response, they almost deserve their own stage. I would call it CDR0. And that's the initial immune detection and response phase, where the TRMs, they dictate how the local immune system reacts, right? So they detect the threat, they respond to the PAMPs and the DAMPs, they shift their phenotype to M1, they start sequestering iron and release the pro-inflammatory cytokines, reactive oxygen species, and basically all of this is signaling immune activation. And then those signals are that basically tells the nearby cells to shut down, or the nearby cells, mitochondria, to shut down OXPHOS and enter CDR1. And so what happens when they become, the TRMs become overactivated? They lose their ability to sequester iron. they don't just lose their ability. It actually, what is it? Ferroportin and hepcidin. I don't know how to say those words. What actually controls the sequestration of iron, that becomes dysregulated, and it starts actively increasing iron export into the local terrain. So it's not just that we can't sequester it anymore. It actually gets flipped backwards, and you start getting more. They experience blunted signaling, making CDR transitions less precise and controlled. And, their continued glycolysis over oxphos increases as well, increasing lactate buildup and reinforcing the metabolic dysfunction. So Mark, in his book and in the cellular suppression theory, describes how iron accumulation and lactate buildup create that terrain advantage for fungi. He mentioned it, he mentioned this, but I'd highlight that increased cell permeability makes intracellular infection more likely. And that's where Mark mentioned that, but since he didn't really have a lot of evidence to say that the fungi needed to be intracellular, he just kind of said, oh, and the cells become more permeable. So in any case, I think Mark's spot on with all of that. The critical point that I would add is that tissue-resident macrophage signaling becomes less precise, therefore making CDR transitions more erratic and more susceptible to external suppression.
It sounds like the entire cancer research should shift towards leveraging the CST, what CST has explained. And do you think if we investigate the ramifications of CST and its effects on local cell, it gives us everything that needed to treat cancer completely? This is where we are at?
Sort of. I want to say yes, but sort of. So the CST finally gets us looking in the right place to truly heal cancer. And honestly, that was Mark's goal all along. That was what he wanted to do. He said, here are all the problems with what we're doing, and it's not getting us anywhere. Start your research here, because this makes perfect sense. That's what he wanted to do. But there are a few more things that we need to address. Knowing what needs to be done, I guess, is one thing, but getting the treatment there is another. Local immune fatigue means the body isn't effectively responding, right? And this is giving the fungi more time to reinforce their defenses. And one of the biggest problems with that, you get a physical barrier, that's biofilm, right? So biofilm creates this physical barrier around tumors, blocking immune cells, antifungal drugs, if that's, if we're going that direction, and oxidative stress. So our own defense from reaching the cancerous cells. That's why many therapies fail, is they can't effectively bypass the biofilm to reach the cancer cell. But so breaking biofilm is just the first step. Your therapeutics, whatever they are, still need to reach the fungi that are inside both the structural cell and the macrophage, specifically the tissue-resident macrophage. And so some drugs like the azoles can enter cells, but they come with significant side effects. And extracellular antifungals might kind of clear surface level infections, and that actually might help some. But the intracellular fungal infection, we require treatments that can penetrate the cell to treat and or kill the fungi inside the cell. The other problems that you run into is you've got chronic inflammation and oxidative stress, right? So they're still creating this self perpetuating cycle. Now, remember, those are damps. And when those are high, then you've got this perpetual damp signal. Remember the damage associated molecular pattern. Sorry, that was hard for me. And that's going to favor fungal survival. So now, even if you can eliminate the fungi you have to conditionally reduce oxidative stress to allow normal immune signaling to reset. And I say conditionally because redox balance is a really delicate thing. Too much reduction and you impair immune function because those signals are actually also the things that are the reactive oxygen species are the things that are killing the pathogens. And if you get rid of too much of this, well, then your signaling is gone and none of the other cells know that they're supposed to be doing something to support and so on and so forth. So you've got to be careful, but if it's done correctly, then reducing oxidative stress in the structural cells will reduce the damps, and reducing it in the tissue resident macrophages can reset their cellular, their cell defense response regulatory functioning, right? Now, at the same time, both the structural cells and the TRM macrophages need sufficient metabolic cofactors and mitochondrial supportive nutrients to fully recover. And local detoxification pathways need to be stimulated to start clearing cellular debris. Then, I feel like I'm rattling off all sorts of things that are needed here. Then, once the cancer cells transition to CDR3, apoptosis resumes. But they can only self-terminate if their apoptotic pathways are intact. So, for example, if P53 function is impaired, even a properly signaled cell with the mitochondria now sending out the signal, it might still fail to self-terminate. So, actually, one of the biggest mistakes I believe one of the biggest mistakes in conventional cancer treatment is the assumption that simply killing the cancer cell is the goal. The problem is that when you aggressively destroy cancer cells, actually any cells, but let's say we're aggressively destroying cancer cells, say with chemotherapy or radiation, or even cytotoxic natural compounds, you risk triggering the production of cancer stem cells, CSCs. They're a subset of cancer cells that are far more resilient, highly adaptable, and resistant to treatment. And when cancer cells are killed too abruptly, the body often responds by activating survival pathways that push some of the remaining cancer cells into a stem cell-like state. And these stem-like cells are they're more invasive, more metastatic, and significantly harder to eliminate. This is one of the reasons why cancer frequently, comes back after treatment. Except that the second time around, it's a lot more aggressive and a lot more resilient to therapy. So a truly effective cancer treatment, you must restore normal cellular function rather than simply wiping out the cancer cells. Because by allowing the cells to progress through the CDR naturally and then reestablishing proper immune and metabolic or mitochondrial function, you avoid pushing cancer into that more dangerous state, right? And this, I'm saying cancer, but the same thing happens for normal cells, right? So this is where cancer cells are acting normal. When you kill them off rapidly, that's a big deal, and something's going wrong, and we need to be able to replace these cells real quickly. So we take some of the cells that are still around, turn them into a stem-like state so that we can heal really quickly. Same thing happens to cancer cells. They're not any different in that state. And so if you've got a way to kill all the cancer cells, well, the ones that you don't kill are going to say, "Oh my goodness, I've got to do something to make sure that we can survive." Because remember, the cells aren't like they're not bad cells. They're following what they're supposed to do. It's just some of their functions are being suppressed a little bit. So that was a lot. Let me run through it real quick. So to treat cancer, completely, we want to prioritize a local treatment. You have to penetrate biofilm, so you can actually access you need to penetrate the cell to eliminate the intracellular fungal infection in both the structural cells and the macrophages. You want to conditionally reduce oxidative stress in both the structure cells and the macrophages to reset that normal immune signaling. You want to provide metabolic cofactors and mitochondrial supportive nutrients to help restore that proper immune and cellular function. Want to provide you want to enhance proper P53 function so that you want to enhance the apoptotic pathways. And lastly, I guess I didn't mention this, all of this, I sort of did. All of this needs to be done in accordance with the body, not in a pathogen fashion. Otherwise, you risk that creating cancer stem cells. That salutogenic lifestyle changes, such as optimizing diet, exercise, and sleep, eliminating toxin exposure, whatever caused the local immune fatigue. Or another way of looking at local immune fatigue is tissue resident macrophage fatigue. Whatever caused that in the first place is all great. But because they're systemic, they're only half the solution. Remember, the first thing I said was you need to figure out how to be able to treat this locally as well.
Yeah. So this is very exciting. This sounds like just theoretically you have cured cancer and you laid out everything, whatever need to address to cure cancer. Of course, assuming everything works the way you have explained. So I guess the question is, why are you so confident that this is how it is going to get cured?
I'm confident because, first off, Mark's cell suppression theory, really, it's already a really compelling framework, right? It lays everything out pretty well. But I'm not confident, or I don't have the confidence level that I have just because it makes sense. I'm confident because all the evidence, supporting it is overwhelming, and some of that evidence isn't widely known. But let's start with what's already been discovered in mainstream cancer research, right? So, even if people haven't connected those dots, you've got fungal signatures are constantly found in cancer. So that the pan-cancer microbiome studies have confirmed that different cancer types have distinct fungal profiles, and we're not just seeing random contamination. Specific fungi consistently show up in tumor microenvironments. And what's more important, I guess, is that their presence correlates with immune suppression as well, exactly as the CST predicts. So if cancer were simply uncontrolled cell division, why would we keep finding fungi species embedded in tumors? And why do they appear to be shaping immune activity, right? So it's not just, this isn't just correlation, right? So these fungi are actually influencing tumor microenvironment. So that's one. Another is, I guess, kind of one of the biggest smoking guns here is most of the drugs that have shown off-label cancer effects, like metformin, ivermectin, statins, and even traditional antifungals like fluconazole, one of the azoles. They have strong antifungal properties. So remember, these drugs weren't designed to treat cancer, yet they seem to work. And what do they all have in common? They disrupt fungal metabolism. And excuse me. So mainstream medicine, even in researching these off-label drugs for cancer use, they try to tie some of the mechanism of action to what the drug is supposed to do. And, so they, some claim to reduce inflammation and others claim to regulate metabolism and others, so on and so forth. But they all adversely affect fungi. And, even ones that shouldn't or weren't ever intended to. If you actually look into it, you find out that, oh, hey, that actually, even aspirin can damage fungi. So all of these things also damage fungi. So again, okay, correlation doesn't cause or it isn't causation, but it's happening every single time. So the other thing is that fungal manipulation of host organisms is actually fairly well documented. So fungi don't just infect, they actually control behavior. And we've seen this repeatedly. There's a particular fungi, for example, that can turn ants into zombies, overriding their neurological function and forcing them to climb to like an optimal spore dispersal height, before it kills them. Or there was a Carolyn Elia at Harvard, and she studied another fungi that hijacks the nervous systems of flies, and it infects the flies so that the infected flies actually take on this specific posture that maximizes spore dispersal. And even after the fly dies, the fungi then manipulate the chemical signals to attract healthy flies to ensure further spread. So, if fungi can hijack an entire organism's nervous system, now it's not obviously not as complex as larger mammals, but is it really a stretch to say that they can suppress CDR progression at the cellular level? It doesn't seem like, right? Mechanisms of fungi used to control behavior and immune function are actually far more sophisticated than people realize. And in truth, what they're doing to suppress CDR progression isn't that complex. Now, another thing is fungi can evade the immune system and persist for really long periods of time. Beyond their ability to manipulate, fungi can also, they're really good at evading immune detection. They can enter the low immunogenic form and effectively hide even inside of a cell. And by shifting into these, call them stealth modes, they don't always trigger an immediate CDR response. So instead, they can actually exist there passively inside the cell until the terrain fully shifts in their behavior from, whatever that carcinogen that is repeatedly activating CDR or activating the CDR, triggering the CDR. And so potentially the fungi can actually just sit inside cells and wait through multiple CDR cycles before actually taking over. And we know that fungi have really highly robust oxidative defenses, and that makes them particularly difficult to clear, especially if they're intracellular. And that suggests that fungal infections and cancer could be silent at first, and then persisting unnoticed until the conditions are optimal for them to suppress immune function and then reinforce the CDR2 survival state. And all of that stuff, we're hacking together a lot of evidence in its favor, or in the favor of the cell suppression theory. But I'm kind of burying the lead again here. Because most researchers study cancer from a theoretical perspective. They're examining genes or metabolic markers and immune responses and isolated lab conditions and stuff. Ionic Alliance Foundation has taken a different approach. For over a decade, we've been actively testing antimicrobial-based approaches to cancer and chronic disease with preclinical efficacy data that supports cell suppression theory, not just for cancer, but for other chronic diseases as well. And here's what makes me so confident. We've never seen our antimicrobial approach fail to show efficacy, regardless of the cancer type or severity. So this really isn't just an interesting theory. We've spent years watching what happens when you remove fungal suppression, no matter the type of cancer, no matter how advanced, we've seen the same results. When you eliminate the fungal driver, the tumor loses its grip. And that's not to say that everyone has always been cured, but we've always seen efficacy. And that's really why I'm so confident in the cell suppression theory, because we've already seen it work.
Wait, so kind of what you're saying is you have been already doing this and all of this, and you have cured cancer for based on your testing and everything.
Well, so I hesitate to go near that statement. First, because if I did, FDA would probably throw me in jail. But second, more importantly, I guess, because cancer is complicated, and throwing around the word cure is dangerous. That said, I genuinely believe that we're getting very, very close. So let's back up. You probably noticed that just about everyone these days has, a cure for cancer. There's lots of hyperbole, and all of these come with, big promises, and they're diet, detoxing, immune stimulation, supplements, whatever, all sorts of different things. Actually, even immunotherapy, and stuff that absolutely can help. For sure. And we've touched on why those things can really help. And in rare cases, some people do actually even reverse cancer with just, say, diet and lifestyle alone, but that's not the norm. And so here's the thing. The most of these interventions aren't wrong. They're just incomplete. And so, we've talked about how mitochondria and metabolic function help. So, of course, a ketogenic diet, fasting, all this can be beneficial. But the idea that cutting out glucose starves cancer, that's just oversimplified. Yes, cancer cells prioritize aerobic glycolysis, meaning there's oxygen around, but we're continuing to stay in glycolysis. We know this. But your body's not stupid. It'll just make glucose from protein if that's what it needs. And, so if you don't eat enough protein, your body will break down your own muscle mass, instead, and convert that into glucose and energy. And actually, sort of inside that, this is where the essential amino acids can kind of come in really handy, because they support tissue repair without fueling glycolysis. And interestingly, studies show that a low protein ketogenic diet combined with the essential amino acids actually helps fight cancer. But so diet alone won't fix everything, right? So an ideal cancer diet is an interesting topic, but complex, right? Mark's book details how late stage cancers adapt when glucose starts to become scarce. Instead of just relying on glycolysis, they start to adapt, and they can use fatty acids. So a keto diet isn't necessarily a cure-all. They can use lactate via the reverse Warburg effect, which is really just making energy out of lactate, and they can use glutamine and arginine. So arginine is actually especially useful, I guess, for cancer, not from the cancer's perspective, because it fuels multiple pathways, right? So it fuels cell proliferation, immune suppression, and nitric oxide production. We've talked about that a little bit as well. And here's actually where it gets interesting, right? So as cancer progresses, it can outgrow its ability to produce arginine, making it oxytropic, meaning that it's completely dependent on the dietary arginine. So arginine is not one of the essential amino acids our body can make it. But if we can't, if the cells can't make it fast enough, well, then those cells, the cancer cells, have to make sure that they're getting it from outside. And that's actually kind of a huge thing, because normal healthy cells can produce all the arginine they need, but the cancer cells can't, because the cancer cells are trying to use that for lots of energy. And So that means by restricting dietary intake, can selectively starve cancer cells while leaving the healthy cells untouched. So a lot of what the ketogenic diet was supposed to do to cancer cells, but with arginine instead of glucose. So that's cool. And to be honest, a ketogenic diet, depending on the protein sources that you use, and that would probably naturally be lower in arginine levels, which could, all of this could be a good thing. And by the way, since arginine is not one of the essential amino acids, if you were on a low protein ketogenic diet and had a significant amount of your protein source come from essential amino acids, well, then you're not getting any of the arginine at all. But you're still getting enough protein that your body can use for building blocks. So that's actually probably why that worked so well. So I apologize for the tangent there. And Mark discusses a lot of this in his book, The Cancer Resolution. So if you're interested in this and want to really understand the dietary inner workings and stuff, I encourage people to go to his book, because he lays a lot of this out really well. My point with the diet discussion there was that rarely are things universally good. Right. So as we kind of talked about, a ketogenic diet isn't universally good for cancer. Honestly, most of the time it's good, but there can be reasons where it's also not going to help much because the cancer cells can also use fatty acids, so on and so forth. But by eliminating intracellular fungal infections, defatiguing the, I don't know if that's a word, I might have just made it up. Revitalizing, that's probably a better word, the tissue resident macrophages, and resetting cellular signaling, supporting optimal mitochondrial function and renaturing P53, those are all universally good. So have we cured cancer? I have to be super careful here. I absolutely think we're on the right track, but cancer is also highly adaptive, as we've kind of touched on, and there's nothing that works for everyone every time. Even if you do all of these things that are really good, that's not to say that it will be a cure. Will it be better? Yeah, of course. You're moving things in the right direction. If you can continue going that way, eventually, hopefully, you'll be cured. Sometimes there are other issues and we can't continue going. So here's what I can say, right? We've developed a local first treatment that bypasses the digestion and systemic delivery entirely. This dramatically increases the local availability of the metabolic cofactors, mitochondrial supporting nutrients, and antimicrobial agents. The localized availability to levels far beyond what's possible or safe systemically. Yet, once those compounds diffuse into circulation, their concentration normalizes pretty quickly. And actually, in the testing that probably wouldn't hold up for the FDA, we've shown that it reduces to baseline pretty quickly, and over a long period of time, we haven't been able to change the systemic levels of the, say, zinc, copper, magnesium, and sulfates that we've got in our product or in our science. I don't want to talk about any products. It absolutely cuts through biofilm, increasing bioavailability and making it easier to access infected or cancer cells, which also means that it's going to help anything else get there as well. It can safely penetrate into the cells to access the intracellular infections. It's one of the most potent antimicrobials we've ever tested. So far, we haven't found a single pathogenic microbe that it doesn't eliminate at relatively low concentrations. It delivers the metabolic cofactors needed for that conditional oxidative stress reduction as fast as the cells can produce that superoxide dismutase enzyme, if you remember that, to regulate reactive oxygen species. It restores mitochondrial function and even helps renature damaged P53. And it may even help repair DNA. all of this is done in a salutogenic fashion to minimize cancer stem cell generation. So if you really dig into the cell danger response, the metabolic theory, and the cell suppression theory, it looks like these are the mechanisms together that you would need to cover kind of everything to treat cancer and, in truth, many chronic diseases. And yes, if you haven't guessed, I'm talking about ion biotechnology, aqueous ligands, or IBAL, IBAL. And again, I want to point out that none of this has been proven to any regulatory body. Officially, IBAL is only recognized as an ingredient, or the science behind the ingredient that is antimicrobial and has how do I say that? Has antimicrobial and antioxidative properties. And this is officially recognized for infection control and wound healing in Mexico. So as long as you're in Mexico, these properties seem better than apparently anyone's ever seen. And so I'm trying to poke a little bit of fun at the FDA there. But I also agree with them somewhat. There's still a lot of unknowns. Even if IBAL works exactly as I suspect, we still have unknowns like dosing, right? And even if we perfected dosing, then we also know that IBAL isn't in an ideal form yet, right? Right now it's topical, which is great for wound care and suppressing, I'll skip that. It's great for wound care, and I guess I'll say it could be surprisingly effective for cancer, even though this product hasn't been used for that, because it allows this local first delivery. But direct injection directly into a tumor would be far better. We know that's pretty straightforward, but that's not something that we can legally do without regulatory proof. And that's where I absolutely agree with the FDA. Don't go injecting something into your body that hasn't undergone that the proper safety testing. And then that kind of ties back to some of what we've discussed in past episodes. This is where our regulatory system really fails patients, right? Salutogenic treatments like IBAL should have far more regulatory leeway than cratogenic ones. And it's frustrating to know that we have a potential breakthrough treatment that could help with cancer and chronic disease, but I can't legally make that claim. And if we could, it'd be much easier to get the funding to run trials, refine the dosing, and start helping people with an injectable form as well. But everything I say about IBAL, or any of the salutogenic treatments that I've discussed, has to be framed for educational purposes only. I can discuss science and observations and data, but I can't say this product or any product treats cancer. And even even though most of the IBAL-based cosmetic products, their sales go to integrative doctors and practitioners, they can't claim anything either. At best, they can frame it like a basic supplement. Providing the right nutrients may help your body heal, and this product may provide nutrients in a way that can't be done otherwise. And so really, as far as the FDA is concerned, we can't even, actually, as far as the FDA is concerned, we can't even claim that IBAL supplies the nutrients, even though we know it does, or at least in Mexico, we know it does. So where's that leave us? We know IBAL can help people. We know it works at a fundamental level. We know it's really, really safe. But the world makes it incredibly difficult to help people, especially when whatever you're trying to help people with is very disruptive. So that's kind of where we're at.
Yep. I like to call this optimism, but cautious optimism here. That's where we are at. But on that note, we will conclude this conversation. Today's big episode. But
I just looked at the time.
Yeah. So once again, thanks for your insights, Jason. And we want all our listeners to listen to this episode, share your thoughts, even reach out to us, reach out to Jason. reach out to us through the show's website. And if you have ideas, help us figure it out, this thing. How can we get this? How can we make it more mainstream if we could? So that's the hope. And it's a long fight and it's a long journey. So any ideas are welcome. Thank you everyone for listening.
Yeah.
Thank
you very much. Thank you.